Hi everyone, and welcome to our video on homeostasis. This is part of our Module 5 series, and it is that first section where we're having a look at obviously all of those different things that we need to be able to control within the body. We're going to be having a look at specification reference 5.1.1c, the principles of homeostasis, in this video. First thing then, what we have is our definition for homeostasis. And what you probably learned back at GCSE is the maintenance of constant internal environment. Now, what we are going to add is a little bit more to this. We are going to have maintenance of a constant internal environment, but we're going to add despite changes in external or internal factors. Because what we're really doing now at A level is adding that little bit more detail, a little bit more understanding to what this process actually is. So as far as those internal and external factors go, we could have obviously the external temperature, but we could just as easily have internal body temperature. Obviously other internal factors, things like your blood glucose concentration, the salts in your blood, water potential, all of these different aspects of the internal environment are also going to affect things happening. And therefore we need to control them and keep them typically within a narrow range. Now, what we actually find as a better definition is going to be the maintenance of a stable internal environment despite changes in external or internal factors. And the reason the word stable is better than constant is because the word constant basically just means that it stays exactly the same. If something is constant, it's kind of unchanging. But the reality is that these conditions within our body, they're not completely static, they will fluctuate. So what we actually find is using that phrase stable does give that little bit of leeway for those fluctuations, but they are being kept within a narrow range. Now I've given you a little exam tip there, because when we have questions asking us about certain things like the internal environment, then a lot of people jump straight in and just say it. That's how you start the answers. Now, it can be very vague, and obviously depending on the question, sometimes that's not going to be good enough to get you the marks, because it could be more than one thing. So at the start of that answer, my best tip to you is always state what it is you're actually talking about. So if we're talking about the internal environment, say the internal environment, don't just say it. Now, what we need to understand then is how this homeostasis is actually brought about in the body. And what we're going to do is lay some groundwork for some of the bits we're going to go into in a lot more detail further on in Module 5. But the key thing that we need to remember here is that there are two key structures required for these response mechanisms to actually work. First part, those sensory receptors. Second part are the effectors. So the sensory receptors are going to be the cells or sensory nerve endings that will be responding to a stimulus in the environment. And this could be internal or external, remember. And the whole idea is that once they've actually detected that stimulus, they will be able to create an action potential. Now, what we're going to find is that we have obviously internal receptors monitoring things like your core body temperature, for example, but we will also have external receptors which are going to be detecting the changes in the external environment, because we've got to be able to respond to both internal and external environmental changes equally. Now, when we've actually got that stimulus causing that sensory receptor to send the signal to the effector, then there's two ways it could get there. The first way is through the neuronal system, and the second is through the hormonal system. So what we're seeing here is there are two different basically communication pathways that the body could employ. And we're going to look at both of those in more detail in future sections of Module 5. When we get to those effectors then, these are going to be the cells, tissues or organs that bring about that response. And typically, if we're talking about some kind of response that requires movement, that would be muscles. If we're talking about something that we're going to see in more detail, like our blood glucose concentration, that could very well involve our liver cells, for example. Now, what we actually looked at back at GCSE is this idea of a negative feedback system. And negative feedback is where we've got a mechanism being used to reverse a change and bring those conditions back to the optimum. So what we're going to see here is that we've got our little change away from the optimum first of all. So something has changed away from where it should be. We've got a receptor that's going to detect that change, and then we're going to send information along the communication system to the effector so that the effector is going to then reverse it. So negative feedback is always about reversing the change and bringing it back to the optimum there. So what we're going to see here then is as those effectors are reversing the change, the system moves closer to the optimum, and as a result of that, the stimulus is going to reduce. Because remember, the stimulus is the moving away from that optimum point. So if what we're doing with our effectors is bringing us back towards the optimum, we're reducing that stimulus, and therefore there's going to be a reduced input to the coordination center, and so that's going to reduce the activity of the effectors. As far as these negative feedback systems go, what they must have in order to be working are: a change to the internal environment must be detected. We've got to be able to send information about that change to other cells. So we've got cell signaling in some way happening here. And then we need some kind of a response to reverse that change in conditions. And obviously, depending on the given context, the bits that we're looking at there in terms of the change, the actual method of signaling and the effector will be different. So you may have to apply that in different contexts, but the overarching principles are the same in every case. What we do need to bear in mind as far as negative feedback systems go is that because we've got this moving away from the optimum and then bringing it back towards the optimum, then what we're going to find here is that we've got this whole principle of overshooting. So it's not a case of we're just going to get to exactly the optimum, job done. We are going to see a little bit of overshoot. So we're going to kind of almost overcompensate a little bit. And it's not immediate. There's always going to be a bit of time taken to respond to whatever that stimulus was. The new bit for A-level is a positive feedback mechanism. So negative feedback mechanisms are where we are then going to bring about a response that's going to reverse the change. A positive feedback mechanism is going to increase the change, moving that system further from the optimum. Now, these are less common than negative feedback systems because quite often what we're going to see is a destabilization of the organism itself, and that's typically quite harmful. So what we see in a positive feedback system then is we've got our optimum here. We see our change away from the optimum, detected by those receptors still, and then we're going to have some kind of communication system signaling the effector, but in this case the effector is going to increase that change. So it's going to keep moving it away from the optimum. If we think about a couple of examples then of positive feedback, as this is our new one. First one, body temperature. If our body temperature has dropped below a certain level, then enzymes are going to become less active because they've obviously got less kinetic energy, therefore less frequent collisions, and then less frequent formation of enzyme substrate complexes. What we find is that because our enzymes are less active, these exergonic reactions are going to be slower. And as a result of that, they're releasing less heat. So this means that the amount of heat our body is generating through reactions is reduced, and so the body is going to cool further. As a result of that, enzyme activity reduces further, and we get this continuing decrease of the temperature. So quite clearly, that's not a good thing for organisms. Generally speaking, we don't want temperatures to keep dropping, and therefore this will frequently have a negative impact on that organism and in all likelihood its chances of survival. Second example I've got for you of positive feedback is good old childbirth. Now, when you are giving birth to a child, then what we've got is a cervix, and the cervix is going to start to stretch. Now, at that point of stretching of the cervix, then oxytocin is going to be released by the posterior pituitary gland. So what we're seeing there, we've got stretching of the cervix, hormone oxytocin is going to be released by that posterior pituitary gland, and the oxytocin is then going to increase the contractions of the uterus. Now, when the uterus contracts more, the cervix is going to be stretched further. As a result of that, we signal the posterior pituitary to release more oxytocin. That then increases the uterine contractions further stretching of the cervix, etc. And basically, this is how we actually get that dilation of the cervix, which then allows a baby to be able to pass through without rather unpleasant moments happening, let's say. So it allows that full dilation of the cervix, the baby can be born, and therefore this is a positive impact of a positive feedback system. So what we can see is different scenarios, some are positive, some have a negative outcome for our organism, but do be mindful of the difference between positive and negative feedback. So negative feedback, we're going to bring it back to the optimum. Positive feedback, what happens with our effectors takes it further from the optimum. I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and of course head on over to the website where you can find an array of other materials to help you in your A-level biology studies.